Welcome to this next video in which we are discussing sarcoidosis. Okay, so we've now discussed the different tissues of the body which are commonly affected by sarcoidosis and which commonly get granulomas forming in them, and the major one is the lungs and the mediastinal lymph nodes, pulmonary sarcoidosis. Okay, so we're about to discuss the pathogenesis of sarcoidosis, what is understood about why this actually happens. But before we move on to that topic, let me just describe a little bit about the course of the disease. So, a lot of cases of sarcoidosis, they actually go into remission on their own within 1 to 3 years. So the statistic is that actually a half of cases of sarcoidosis, so 50% of people with sarcoidosis will go into remission. Okay, so the disease will remit, it will go away, everything will get better on its own within 1 to 3 years. So within 1 to 3 years. So it's not very well understood why this actually happens, but this is a fact about the course of the disease, that about half of people with sarcoidosis will get better, it will go away within 1 to 3 years. Okay, now, another statistic, about 2/3 of cases of sarcoidosis will go into remission after around 10 years. So after 10 years, 2/3 of cases of sarcoidosis will have actually gone into remission. So this is counting the half that went into remission within 1 to 3 years. So on top of that, another, so let's just get them over common denominators. So this is 3/6, this is 4/6. So another 1/6 of people will eventually get into remission by 10 years. Okay, so after 10 years, 2/3 of people with sarcoidosis will have got better, the disease will be in remission. The remaining 1/3, unfortunately, the disease can go on and on and on for decades. Okay, and it's not very well understood why some people get better from the sarcoidosis whilst others it goes on and on and on. We will discuss theories about this later on. Okay, so now what we're going to discuss is the pathogenesis of sarcoidosis. So the general theory is that most people agree that sarcoidosis occurs in people who are genetically susceptible, who are exposed to a certain environmental agent. So it's believed that you need to be genetically susceptible. So it's believed that not everyone can actually develop sarcoidosis. So there are some people who we refer to as genetically susceptible hosts. Okay, so certain people are capable of actually developing sarcoidosis is the general theory that most people agree on. So there are genetically susceptible people to develop sarcoidosis. However, to actually develop the sarcoidosis, they have to then be exposed to a certain environmental agent. So there is something that they have to be exposed to, and then because they're genetically susceptible and they've now been exposed to the correct agent, they'll then develop sarcoidosis. So now they will get sarcoidosis. So it's not solely genetic and it's not solely environmental. So not everyone can actually develop sarcoidosis is generally what people agree on. Only people who are genetically susceptible to it, but genetic susceptibility isn't enough to actually get the disease. You have to then be exposed to the correct agents that you are susceptible to. Okay, so this is the general agreed upon theory of how sarcoidosis develops. You are genetically susceptible to it, but you then have to be exposed to the triggering agent, and then you actually get sarcoidosis. Okay, so that's all I want to say at the moment about the pathogenesis of sarcoidosis. What I now want to do, to understand why these granulomas actually form, is go to a very much so related disease, but a different disease, or at least, well, as we'll see later on, there's a theory that actually they're not different diseases, but we're going to go to a disease that is certainly very similar to sarcoidosis, and we're going to study that disease, which is better understood than sarcoidosis. It's also a systemic granulomatous disease, and it is, of course, tuberculosis. So we're going to study tuberculosis. We're going to have a huge interval now where we're actually going to just study tuberculosis, and the reason is that if you want to understand sarcoidosis, really you have to look at tuberculosis. Tuberculosis is so similar to sarcoidosis, and by studying tuberculosis we can get insight into how granulomas form and why they form, what the purpose of them is. And I really think that it is valuable to look at the pathogenesis of tuberculosis if you want to understand sarcoidosis. So we're now going to have a very long interval then where we're going to study tuberculosis rather than sarcoidosis, but it will absolutely be worth it. This will give us an understanding of what is happening in this disease, and indeed later on we'll see that one of the great theories of sarcoidosis, of the cause of sarcoidosis, is that actually it is just tuberculosis. It's just part of tuberculosis, it's part of the spectrum of what tuberculosis can actually cause, but that's still controversial. That's known as the mycobacterial theory of sarcoidosis. So just to make sure that everyone's absolutely clear with where this conversation is going, we're going to now have a great long discussion of what happens in tuberculosis, also known as TB. And we're going to see that in TB you also get granulomas forming around the body, particularly in the lungs, but they can form all over the body. We're going to, from this discussion, understand how granulomas form and why they form, what the purpose of a granuloma is. Then what we'll do is we'll go back to sarcoidosis and we'll use what we've learnt about tuberculosis to fill in now some of the gaps in our understanding of what actually happens in sarcoidosis. And then finally we'll finish up by discussing the two major theories of the etiology of sarcoidosis, which are the mycobacterial theory, which says that actually sarcoidosis is just part of the spectrum of tuberculosis. And then we'll also look at a very different theory, which is the propionibacterium acnes theory, which says no, that's rubbish, it's caused by the same bacterium that causes acne. Okay, so I should say actually right now that the pathogenesis of sarcoidosis and what causes sarcoidosis is still officially unknown. It's still very controversial, and the evidence at the moment doesn't point really clearly in any direction. So there are lots of different theories. We're going to discuss the two most famous theories, which are the mycobacterial theory and the propionibacterium acnes theory later on. But at the moment we're going to study tuberculosis to give us an understanding of how granulomas actually form. Okay, so I hope it's clear where this conversation is actually going. So now we're going to have a long interval where we're going to discuss tuberculosis. So, tuberculosis then, TB. So, we'll start off with the causative agent of tuberculosis. What is it that causes tuberculosis? Well, it is majorly the bacterium by the name of Mycobacterium tuberculosis. So the major cause of TB is this bacterium, Mycobacterium tuberculosis, and becoming infected by this bacterium. And for short, we'll often be abbreviating Mycobacterium tuberculosis down to MTB for short. M for Mycobacterium, and TB is then for tuberculosis. Okay, so let me begin with a little bit of bacteriology then. Let me describe Mycobacterium tuberculosis to you. So Mycobacterium tuberculosis, it's a small bacillus, okay, which means that it's rod-shaped. So in bacteriology, the word bacillus refers to a rod-shaped bacteria. So Mycobacterium tuberculosis is a bacillus, and in fact, the word tuberculosis actually comes from the combination of the word tubercle and bacillus. So tuberculosis is short for tubercle bacillus, and the reason that we've got the word tubercle in here is because in tuberculosis you're going to get granulomas forming, which are tubercles. Okay, another word for a granuloma that's resulting because of TB is to call it a tubercle. So the tubercle forming bacillus, tuberculosis. Okay, so Mycobacterium tuberculosis, it's a bacillus. It's a non-motile bacillus. Now this means that it doesn't have any flagella, so it's not capable of actually propelling itself around. It's not capable of actually moving. Now, it has a cell wall around the cell membrane. So this line here, this is supposed to represent the cell membrane of the Mycobacterium tuberculosis. Surrounding the cell membrane, it's going to have a cell wall. And the structure of this cell wall is quite complicated. It's very different from the structure of the cell wall of most bacterial species that famously cause disease in humans. So Mycobacterium tuberculosis is neither gram-positive nor gram-negative, and the reason is that it just doesn't stain if you throw on the gram stains onto it. So the gram staining process is a process used to stain many bacterial species, and bacteria stained through the gram staining process can either show up blue, in which case they are called gram-positive, or they can show up red, in which case they're called gram-negative. Now, Mycobacterium tuberculosis, if you put it through the gram staining process, it just does not work. It doesn't stain at all with gram staining processes, so it's neither gram-positive nor gram-negative. And the reason is that its cell wall contains a lot of mycolic acid molecules. And I should say that the portion of the bacterium that stains with the gram staining process is the cell wall. So it's the cell wall that actually takes up the dye molecules. And the presence of these mycolic acid molecules in the cell wall of Mycobacterium tuberculosis prevents it actually taking up the gram staining dye molecules. So it doesn't stain with gram staining processes. Instead, we have a special stain to stain Mycobacterium tuberculosis with, which is known as the Nielsen staining process. And this makes the bacterium show up as a bright red rod. Okay, so Nielsen staining process is the process that you can use to stain it with. So we would refer to Mycobacterium tuberculosis as neither gram-positive nor gram-negative. Instead, we'd say that it stains with the Nielsen stain process. Okay, right, so it's got this cell wall that contains a lot of mycolic acid molecules. It's very different from normal cell walls of bacteria, and this is the reason that it doesn't stain with the gram staining process, and we need this special stain called the Nielsen stain. The one final thing that I want to tell you about Mycobacterium tuberculosis cells is that they have a single circular chromosome, like so. Okay, so bacterial cells, they also have chromosomes. Unlike human cells, bacterial chromosomes are often circular rather than linear. Do not confuse this with plasmids. Plasmids are tiny little circles of DNA that bacterial cells can have. This is an actual chromosome. This is a massive, great loop of DNA. So the Mycobacterium tuberculosis, it has a single chromosome which contains all of its major genes. It can also, of course, have a few little plasmids which will contain a few side genes that it can also use. Okay, right, so there's a description of the Mycobacterium tuberculosis cell. This is the major bacterium that can cause the disease tuberculosis. However, I should mention that there are a few other species of bacteria very closely related to Mycobacterium tuberculosis that can also cause tuberculosis. They're also mycobacterial species, so their name will also involve mycobacterium, but then instead of having tuberculosis after it, they'll have some other name there. So they'll be different species but very closely related to tuberculosis. And overall, we refer to the collection of bacterial species, mycobacterial species, that can cause tuberculosis as the Mycobacterium tuberculosis complex. So this means all of the different mycobacterial species that can actually cause the disease tuberculosis. However, the major one is Mycobacterium tuberculosis. Most cases of tuberculosis will be caused by the bacterial species Mycobacterium tuberculosis itself, rather than one of the other species in the Mycobacterium tuberculosis complex. However, you should still be aware that some of the other species of mycobacteria, very much so related to Mycobacterium tuberculosis, can actually cause TB. Okay, but we're going to focus just on Mycobacterium tuberculosis because it is the major cause of TB. Okay, so we've studied now the bacterial cell. Let's now actually begin the disease process of TB, beginning with how do you catch TB? How is it transmitted? So we'll begin the story of the disease with transmission. So this requires two people. Person 1, who is infected with tuberculosis and has currently got an active tuberculosis infection. Now, this is a difficult concept. The concept of an active TB infection and the concept of a latent TB infection. This is a concept that is going to be absolutely crucial for us to understand, but it's something that we're only going to be able to understand once we've actually completed up the story. So I'm going to introduce these terms right now, active TB and latent TB, but I'm not going to explain what they actually mean in full detail. I'm just going to tell you at the moment that if you have an active TB infection, you can actually transmit the bacteria to other people, whereas if you have a latent TB infection, you are not infectious. You cannot transmit it to other people. The other difference is that if you have an active TB infection, you're actually usually very, very unwell, whereas if you have a latent TB infection, you're absolutely fine. And when we say that a third of the human population is currently infected with tuberculosis, and it is something like that, around a third of the human population is currently infected with Mycobacterium tuberculosis or one of the very closely related species, most of those people are infected with a latent TB infection. They are not actually ill and they cannot transmit it to other people. Very few of them will actually have active TB, and those that do will be extremely unwell and will be capable of transmitting it to other people. So, person 1 here is going to have an active TB infection because they're going to transmit it to person 2. So person 1 has an active TB infection. So, active TB infection. And TB nearly always affects the lungs, especially if it's caused by Mycobacterium tuberculosis, which is the type that we're focusing on. So, person 1 will be coughing up bacteria from their lungs then. So, when they've got the active TB infection, that will be causing a lot of irritation to the lungs, and they'll therefore be coughing a lot, and when they cough, lots of the bacteria will get expelled into the air around them. Okay, so person 1 has coughed and coughed and coughed, and now there are lots of Mycobacterium tuberculosis cells floating around the place in tiny little aerosols in the air. So here, this is meant to be showing us a Mycobacterium tuberculosis cell. So that's why I've coloured it in red here. Now let's say that we have person 2 here. Okay, so person 2. Now, let's say that person 2 has never ever been infected with Mycobacterium tuberculosis before. So they don't have an active TB infection, neither do they have a latent TB infection. They have never ever been infected with Mycobacterium tuberculosis before. So this is a naive person. We can call them a naive host. They've never been exposed to Mycobacterium tuberculosis before. And actually I should just say that most people, when they've been infected with Mycobacterium tuberculosis for the first time, so person 2 here, who's about to breathe in the Mycobacterium tuberculosis, once you've been infected with it, you nearly always develop a latent TB infection. Very few people actually manage to completely clear the bacteria from their body. Most people who have been exposed to TB bacterial cells will have the bacterial cells inside their body for the rest of their life. They will have a latent TB infection. That's how a third of the world's population is currently infected with TB. But as I say, we'll develop the concept of active and latent TB much more later on. So, this is someone who currently does not have a latent TB infection. They have never been exposed to the bacterium before, and now they're going to breathe in loads of Mycobacterium tuberculosis cells from the air, which have been put there by person 1 who currently has an active TB infection. Okay, so now what's going to happen? Well, of course, the bacterium is going to go down the trachea, down the bronchi, and into the lungs. So I'll just draw a little flow chart of what's going to happen. So here's our picture of the trachea and the bronchi. So the bacterial cells will come down the trachea, down the bronchi, and of course you're unlikely just to breathe in one, you're most likely going to get a lot of them, so they'll be going all over the place, and then they'll go down smaller and smaller airways until eventually they'll end up in alveolar ducts. And let me just move this up a little bit. So eventually they'll end up in alveolar ducts, and alveolar ducts are tiny little airways where the walls of the airway is made up of alveoli. So let me draw this here. So this is what an alveolar duct would look like. You can see that the walls of this airway are made up of these alveolar air spaces here. Okay, so this is a picture of a tiny little airway in the lung known as an alveolar duct. And each one of these little spaces here, this is an alveolus. Okay, so the Mycobacterium tuberculosis cells, they are going to come down the airways until eventually they get into alveolar ducts, and then from the alveolar ducts they're then going to get into the alveoli. And we now want to see what is the body's response to the presence of the Mycobacterium tuberculosis in the alveoli. Because the body does not like the presence of Mycobacterium tuberculosis in the alveoli, and we need to now see what's going to actually happen. So for this purpose, I want to draw a bigger picture of an alveolus, but I think I'll get a new piece of paper to actually do this. Okay, so here's a fresh piece of paper, and now what I'm going to draw is I'm going to draw the side of an alveolar duct where I'm going to have a few alveoli shown on this picture. So I'll put this here, like so. So here's one alveolus, and just to complete the picture up, here's another alveolus. So the alveolar duct would be here, which is the central airway that has the alveoli coming off the side of it. Okay, so what then is the lining of the alveoli made up of? Well, it's made up of pneumocytes, cells of the lung, and there are two major types of pneumocytes, which I'm now drawing on here. There are the type 1 pneumocytes, which are these ones that I've drawn so far, and then there are a few type 2 pneumocytes interdispersed amongst the type 1 pneumocytes. So the type 1 pneumocytes are the really thin ones here, the squamous cells, and these are the ones which are going to provide the very short diffusion distance for gas exchange from the alveolar airspace into the blood. And then the type 2 pneumocytes are these much taller ones here. So, just to complete this picture up, let me put a basement membrane. So all of these cells will be sitting on a basement membrane. This is what keeps them in position here. So in red, this is representing the basement membrane that they're all sitting on. So I'll firstly just label that up. So this is the basement membrane of the alveolar wall. Now let's label up the two different types of pneumocyte that I've described to you. So here, this in orange is an example of a type 1 pneumocyte. And these, as I've said, are the major type of cell that makes up most of the surface area of the alveoli walls, and their purpose is to make a very thin barrier between the alveolar space and the space underneath the alveolar wall, which is where the blood vessels will be, and that's obviously to allow as short a diffusion distance as possible for gas exchange to occur. Okay, so that's type 1 pneumocyte. Now if your respiratory physiology is good, you'll remember then what the function of type 2 pneumocytes is. So type 2 pneumocytes are these odd ones that are much taller, and they make up much less of the surface area of the alveolar air spaces. So this is a type 2 pneumocyte here. Now their function is in secreting the surfactant, which will cover the surface of the alveolar wall, and this is involved, remember, in reducing the surface tension to try and stop the alveolar air spaces collapsing in on themselves. So remember, the surface of these alveoli is reasonably damp. Okay, there's a sort of layer of fluid on the surface of the pneumocytes here. Now if you didn't have surfactant in that surface fluid, what would want to happen is you'd want to press where shall I show it? I'll do it here. You'd want to press this side up against this side. It would be like where if you've ever got two glass slides, if you've ever been doing microscopy and you've got two of those glass slides that you use to look at, well, you use to put your slice of tissue on, okay? If you get two of those and put the two slides one on top of each other, so I'll just draw what I'm showing here, what I'm talking about here. So if you get two glass slides from light microscopy and you put them on top of one another, if they're not wet, then you can easily then pull them apart. If, however, these two glass slides were wet when you put them together in this way, then you will find it really difficult to pull them apart. That is what is meant by surface tension. A very similar thing happens here with the surfaces of the alveoli here. This is wet, this is wet. If they were to press against one another, they then want to stick together rather than being pulled apart. Surfactant prevents them from doing that. It prevents the surface tension, as we say, and it stops the alveolar air spaces from collapsing on themselves and keeps them nicely open. So the type 2 pneumocytes are very important for creating surfactant. Okay, right. So what else do I want to put on this picture? So of course, in this space underneath the basement membrane, you'll then have the microvasculature. You'll have pulmonary capillaries. So I'll show one of these here maybe. So here is a pulmonary capillary, and gas exchange will be occurring between the blood that's flowing through this pulmonary capillary and the alveolar air spaces. So oxygen will be coming in, carbon dioxide will be coming out, and that's the purpose of the type 1 pneumocytes, to keep this diffusion distance as short as possible. So the diffusion distance will overall consist of the type 1 pneumocyte, the basement membrane here of the alveolar wall, then the basement membrane of the pulmonary capillary, then the endothelial cell of the pulmonary capillary, but that's it. And the endothelial cell of the pulmonary capillary will be very squamous and thin, just like the type 1 pneumocyte. Okay, right, so back to our story then. Mycobacterium tuberculosis will be coming into the alveoli then now. So let's have a Mycobacterium tuberculosis cell in our alveoli. So, what's going to happen now? Well, there are cells in the alveoli that are designed to deal with this situation. When bacterial cells come into alveoli, there are macrophages in the alveoli which are there to actually clear any bacterial cells which come into the alveoli. So I'm just going to now draw one of these. So wandering around in between the alveoli of the lungs, there are these giants of cells known as macrophages. So this cell here is a macrophage, and of course we've met macrophages before when we were describing a granuloma. So this is what we would call an alveolar macrophage. So inside your lungs there are loads of these alveolar macrophages, and they are wandering around in the alveolar ducts. They move from alveolus to alveolus to alveolus. So they're not static at all, they move around and they can move in between the different alveoli. And what is their job here? Their job here is to keep the alveoli free of bad stuff, free of any dirt that has managed to find its way into the alveoli, and free of any bacteria and other pathogens that have managed to find their way into the alveoli. And the way that they actually do this is by engulfing bacteria or other pathogens or pieces of dirt, any sort of debris that has ended up in the alveoli, and breaking it down, a process known as phagocytosis, which we will discuss more in a moment. Okay, so these are going to be participating in phagocytosis. There is another type of cell that I just want to mention which can also participate in phagocytosis. It's not going to do it as quite as often as the alveolar macrophages, but it is also important to mention, which is the alveolar dendritic cells. Now these are not wandering around in between the different alveoli, these are actually underneath the alveolar wall here. So I'm just going to show one of these, so I'm figuring out where to put it. I might put it, where should I put it? I'll put it here. So dendritic cells, the reason they're called dendritic cells is that they have these dendrite-like processes, okay, like so, and the reason they have these is so that they can extend into all the different crevices of the tissue, and this really does fit their actual function, which is they are involved in looking for the presence of pathogens. Okay, so this is the reason they have all of these processes stretching around looking for pathogens. Okay, so this is an alveolar dendritic cell, and as you can see, these are underneath the alveolar wall, but I hope what I've shown you with this picture is that some of the alveolar processes will extend through the wall of the alveoli. Okay, you can see that the process can extend up so that a surface of the process, one of these dendrites here, so these processes are known as dendrites. Now, they have absolutely nothing to do with the dendrites of neurons. These are not capable of conducting action potentials. They simply don't have the appropriate voltage-gated ion channels for doing that. Dendrites are just processes coming off a cell like this, and you can see that this cell looks reasonably like the head of a nerve cell. Okay, but with a very different function. In the case of nerve cells, they are collecting information from other nerve cells, and that's the reason they have all of these processes, so that they can collect information from all sorts of different cells. In the case of these dendritic cells, they're looking for evidence of pathogens. So some of the dendrites of the alveolar dendritic cell will stretch through the alveolar wall here, so that they can actually look at what is present in the alveoli, and these can also phagocytose pathogens and dirt and things like that that are in the alveoli. But the major ones that are going to do it is the alveolar macrophages. Okay, so these are two very important cells that are all the time looking for the presence of pathogens inside the alveoli, and if they find it, they're going to phagocytose it, and they're also going to set off the alarm. So what I now want to actually talk about is how are they going to recognize the presence of Mycobacterium tuberculosis? How do they actually decide that this cell here is a pathogen, and then what is their actual response to that going to be? Okay, so the alveolar macrophages and the alveolar dendritic cells, I'm just going to introduce a piece of terminology that summarizes both of these cell types, which is the piece of terminology that they are called sentinel cells. So we can describe alveolar macrophages and alveolar dendritic cells under this umbrella term of sentinel cells. And the reason they're called this is, if you think about what their function is to guard the alveoli. They're continuously standing on guard, looking for the presence of something very bad. And if they find something bad, such as Mycobacterium tuberculosis in the alveoli, they're going to set off the alarm. So they are effectively on guard, which is why they're called sentinel cells. Okay, so we can describe the alveolar dendritic cells and the alveolar macrophages then as sentinel cells. Okay, so now, how then are they going to recognize the presence of Mycobacterium tuberculosis? That's the first question that we need to ask. So the way in which these sentinel cells recognize Mycobacterium tuberculosis, and indeed other pathogens, is by the presence of molecules on the surface of the bacterial cell that would never ever be on the surface of a human cell. So if we think about Mycobacterium tuberculosis, just drawing it here, it's a very different cell to human cells, and it can produce all sorts of different molecules that no human cell in its right mind would ever produce. These are giveaway signs that this cell is not a human cell, and these are the sorts of things that the sentinel cells can actually recognize. They can have receptors for these molecules, and if those receptors get activated, then that's a giveaway sign that we're dealing with something that is non-human and potentially dangerous. Okay, so there's some fancy terminology for this very basic concept. So if I just draw one of these alveolar macrophages here, and by the way, this picture is a little bit out of scale. The bacterial cell would in reality be absolutely tiny compared to the macrophage here. Okay, so here's the macrophage. So, on the surface of the bacterial cell, we've said that there can be these molecules that no human cell in its right mind would ever have on its surface. The fancy piece of terminology that we use to describe all of these molecules is PAMPs, and this stands for Pathogen-Associated Molecular Patterns. So the P is for pathogen, the A is for associated, and the MP is for molecular pattern. So pathogen-associated molecular patterns. So this is an umbrella term for all of these different molecules that loads of different pathogens could have on their surface, which would never ever be produced by a human cell, and which is a giveaway sign that this cell is non-human and potentially dangerous. So this is what we mean by pathogen-associated molecular patterns, or PAMPs. Okay, and on the surface of the sentinel cells, on the surface of the alveolar macrophages and alveolar dendritic cells, we can have receptors for these pathogen-associated molecular patterns, which is what I've shown here, and the umbrella term for these is to refer to them as pattern recognition receptors, or PRRs for short. So pattern recognition receptors, which can be abbreviated down to PRRs. Okay, so I'll colour in the pattern recognition receptor here in green. Okay, so these I want to stress are umbrella terms. PAMPs is the umbrella term for all of these different molecules that we could have on loads of different pathogens, which are the giveaway sign that there is a non-human, potentially dangerous cell present in the tissue. PRRs, pattern recognition receptors, is the umbrella term for all these different receptors for all these different PAMPs, which will activate signaling cascades if they find their ligand inside the sentinel cell to initiate the process of phagocytosis and also the production of alarm signals. Okay, so just to absolutely nail this point home, let me give you an example of a PAMP and its pattern recognition receptor. So, for Mycobacterium tuberculosis, there is a molecule known as lipomannan, okay, which will abbreviate down to LM. So this stands for lipomannan. So this is another molecule along with mycolic acid that is present in the cell wall of Mycobacterium tuberculosis. So it's right on the surface of the Mycobacterium tuberculosis cells. And this is a molecule that no human cell would ever in its right mind make. So it's a giveaway sign that this cell is not human. And indeed the sentinel cells, the alveolar macrophages and the alveolar dendritic cells, they have a receptor on their surface, the ligand for which is lipomannan. And when lipomannan is present on the surface of a cell that they come into contact with, it will bind to these receptors, activate signaling cascades inside the sentinel cell, and cause the sentinel cell to recognize and react to the presence of the pathogen. And the name of the specific receptor, the specific pattern recognition receptor that recognizes lipomannan, is TLR2, which stands for toll-like receptor 2. So T is for toll, L is for like, and then R is for receptor, so the toll-like receptor 2. So that's just a specific example here of a PAMP and its pattern recognition receptor. The key thing that I want to get across with this example is that there is not just one pattern recognition receptor which recognizes all the pathogen-associated molecular patterns. That would be very difficult to build. Instead, there are loads of different pattern recognition receptors for all of the different pathogen-associated molecular patterns, and these different pattern recognition receptors are on the surface of our sentinel cells, and this is the way in which the sentinel cells recognize the presence of the pathogen. Okay, so let's summarize where we've got to in the story so far. So we have breathed in the Mycobacterium tuberculosis. It's gone all over our lungs. We've got Mycobacterium tuberculosis cells in alveoli all over our lungs. And what is now going to happen? The alveolar dendritic cells and the alveolar macrophages are going to recognize that this is not a human cell. And we now want to have a look at what those alveolar macrophages and alveolar dendritic cells are going to do. So one of the key things that they're going to do is they're going to phagocytose it. They're going to try and destroy it. So phagocytosis is the next thing that I want to discuss. And I want to discuss how this is going to fail in most cases. Because Mycobacterium tuberculosis, I'm afraid, has a huge number of tricks up its sleeve to prevent alveolar macrophages and alveolar dendritic cells from actually being able to digest it. So let me discuss this. So firstly, I'm going to tell you what hopefully will happen when a alveolar macrophage or an alveolar dendritic cell attempts to phagocytose a Mycobacterium tuberculosis cell. Then I'll go over the reality in most cases. So this is what should happen. So here is, let's say, the alveolar macrophage. What will happen is it will start to invaginate its membrane around the Mycobacterium tuberculosis. So here, again, drawn completely out of scale, there is the Mycobacterium tuberculosis cell. So here is our alveolar macrophage nucleus, and there's the Mycobacterium tuberculosis cell. So we're invaginating our membrane around the Mycobacterium tuberculosis cell, and what will happen is this will just continue and continue and continue, and eventually will pinch off the membrane. So what this will go over to is looking like this. Here is our alveolar macrophage, and now inside this special vacuole that we've got here, we now have the Mycobacterium tuberculosis cell. So I'll just colour him in again in red. And this special vacuole that we have inside the alveolar macrophage with the Mycobacterium tuberculosis cell inside, this is known as a phagosome. Okay, so that's the first part of phagocytosis, the engulfment process. The alveolar macrophage or the alveolar dendritic cell will take the cell that it's trying to phagocytose, and this is a much more general process than just occurring in tuberculosis. We're doing it, of course, in the context of tuberculosis, but this happens to many different bacterial cells. Okay, you engulf the bacterial cell into a phagosome, and now, of course, what we need to do is destroy the bacterial cell. So how do we actually start to digest the bacterial cell? Well, this, as I say, is what should happen. We'll talk about what actually happens often in the case of TB later on. So what should happen is that you should fuse loads of other little vesicles known as lysosomes with the phagosome. So these little membrane-bound structures here, these are representing lysosomes, and these are something that the phagocytes, these macrophages and dendritic cells, and indeed all cells of the body actually have, but these phagocytes are going to have a special use for these. So let me just draw one of these lysosomes much larger. So it's a membrane-bound vesicle inside a cell which is absolutely full of enzymes that are going to break down biological molecules. So here, these are representing enzymes, and the way that we can summarize all of these enzymes is to call them lytic enzymes. So lytic means to break down. So these are enzymes that are going to break down all sorts of biological molecules. So you'll have enzymes in there that can break down proteins, enzymes that can break down carbohydrates, enzymes that can break down nucleic acids, enzymes that can break down lipids. You'll have loads of different types of enzymes that can break down all of the different biomolecules that you can think of. Now, what are the purposes of these potentially incredibly dangerous structures? Oh, I'm sorry about this. I didn't realise it wasn't in view. Let me just raise it up. Okay, so what then is the purpose of these potentially incredibly dangerous structures inside cells? Well, the major thing that most cells use these lysosomes for is if they have a molecule that they no longer need, they can send it to the lysosome to be broken down into its constituent pieces. So lysosomes are often used for recycling biological molecules inside of cells. However, in the case of our alveolar macrophages and alveolar dendritic cells, they are going to have a more aggressive use for these structures. They are going to fuse these structures with the phagosome here, okay, and that will release all of the lytic enzymes inside lysosomes into the phagosome. And once that process has occurred, the larger structure that you then end up with, which I'll draw here, which will now contain our bacterial cell along with all of these different lytic enzymes here, okay, shown again completely out of scale, but I hope it gets the point across. So here's the bacterial cell, and this is meant to represent a lytic enzyme. This structure is now known as a phagolysosome. So this is a phagolysosome. Okay, so we fuse lysosomes with the phagosome to create a phagolysosome, and hopefully those lytic enzymes are then going to break down the bacterial cell. So that's one of the mechanisms that we use for the breakdown of the bacterial cell that is inside of the phagosome. The other major mechanism that we use is we try and poison the bacterial cell by creating a huge number of reactive oxygen species and reactive nitrogen species in the phagosome here. So let me just go over this. So the fusion of the lysosomes with the phagosome is one of the killing mechanisms. So I'll just put a 1 here. Another killing mechanism is the production of reactive oxygen species and reactive nitrogen species, RNS. So ROS will start off with. So ROS stands for reactive oxygen species. Okay, so the principle here is that we're going to create a bunch of very reactive molecules that react with all sorts of other molecules in a pretty much indiscriminatory way. They're very, very dangerous molecules, and the idea is that if we create all of these molecules in the phagolysosome here, they're going to react with all of the molecules of the bacterial cell and hopefully kill the bacterial cell. So we're just going to create overall a bunch of molecules that are toxic to life, cytotoxic molecules, reactive oxygen species and reactive nitrogen species, and hopefully poison the bacterial cell. So let me start off with the production of reactive oxygen species. So, to produce reactive oxygen species, the first one that you're going to produce is a molecule called superoxide. So let me just discuss where the superoxide is actually going to come from. So in the membrane of the phagolysosome, and I just realised I could have actually used that picture, but never mind, I'll redraw it here. So here is the phagolysosome, which contains our bacterial cell, which in this case we're thinking about mycobacterium tuberculosis still. Okay, so in the membrane of the phagolysosome, what we can construct is an enzyme complex to create superoxide. Okay, so this is known as Nadph Oxidase, also called phagocyte oxidase. So in the phagolysosome membrane, we construct this structure called Nadph or phagocyte oxidase, and this is constructed out of a huge number of different proteins that we have to join together to make the full enzyme complex. Okay, now a lot of these proteins are in the cell membrane, okay, but you don't have all of them in the cell membrane. So, starting again, the Nadph or phagocyte oxidase, it's a protein complex made out of a lot of different proteins. And a lot of the proteins that we're going to use to construct this from are in the cell membrane. So when you invaginate the cell membrane and create the phagosome, those proteins are going to be in the membrane of the phagosome. The other ones that you need to create the Nadph oxidase come from the cytoplasm, and those are now only going to join with the ones in the phagosome, and they're not going to join with the ones that are still in the cell membrane. And in this way, we only get Nadph or phagocyte oxidase complexes being assembled in the membrane of the phagosome rather than in the cell membrane itself. Okay, so I hope that makes sense. So we're going to create this structure called Nadph oxidase in the phagolysosome membrane. Okay, and it's a complex of a huge number of different proteins. This enzyme complex is going to start actually producing a very dangerous molecule known as superoxide. So it's going to take dioxygen molecules, O2 here. So the fancy word for this is to call it a dioxygen molecule, and it's going to convert them into what is known as superoxide molecules, which are written as O2 negative dot So it's going to convert them into superoxide. And superoxide is a reactive oxygen species. It's a very reactive molecule that's going to be very poisonous to cells. It's also going to be a molecule that we're going to use to create a lot of the other reactive oxygen species. So not only is it a reactive oxygen species itself, but it's also a molecule that we're going to use to create other reactive oxygen species. So let me just go over how you actually convert a dioxygen molecule into a superoxide molecule. Okay, so here we have the two oxygen atoms of a dioxygen molecule, and I just want to draw an electron dot cross diagram for the dioxygen molecule. Okay, so remembering basic chemistry then, each of the oxygen atoms has 6 outer shell electrons. So we'll colour the electrons of this oxygen in red. So you're going to have 2 lone pairs of electrons here, and then 1 electron in each of these double bonds here will come from this oxygen. And then the other oxygen's electrons will colour in blue here, so again you'll have 2 lone pairs of electrons, and then it will put an electron into each of these bonds of the double bond here. Okay, so this is the electronic diagram for a dioxygen molecule. To convert a dioxygen molecule, which is very stable, into a superoxide molecule, which is not very stable at all, what you have to do is put in another electron. You have to put into this dioxygen molecule another electron. So here comes this other electron, and what's now going to happen is, in order for this molecule to accept another electron, one of these bonds of the double bond has to break. So let's say it's this one that's going to break, and what happens is one of the electrons the electrons go back to their 2 oxygens. So one electron of this bond will go to this oxygen and one will go to this oxygen, and then one of the oxygens will get to accept the additional electron. So what you'll end up with is something that now looks like this. Here are the 2 oxygen atoms. We've now got a single bond between the 2. So here's that electron in red, here's the electron in blue, so that's that bond there. And now let's say that it's the red oxygen which accepted the orange electron. What it will now have is these 3 lone pairs of electrons, so it has accepted the orange electron in here. Okay, so this red electron came from here, the orange electron was the one that it accepted, and now let's think about the blue oxygen on the other side. It's now got 2 lone pairs of electrons and, oh dear, an unpaired electron. Okay, and this is what makes the molecule so reactive. One side of this molecule has a negative charge, the other side of the molecule has a free radical. Okay, so this is a free radical over here. Whenever you have a molecule that has an unpaired electron, that is called a free radical, and it is incredibly reactive. Having an unpaired electron makes you an incredibly unstable molecule. You react with all sorts of things. And this is the reason this is written O2 negative dot. The negative pertains to the negative charge over here. The dot refers to the free radical. The unpaired electron is what you're showing there. This is an incredibly reactive molecule. Okay, so superoxide is going to be created by the NADPH or phagocyte oxidase. Superoxide is a reactive oxygen species in its own right, but it can also create other very reactive molecules that are based on oxygen, which are also known as reactive oxygen species. So spontaneously it will react with hydrogen atoms to create hydrogen peroxide. So the structure of hydrogen peroxide is like so. It's H2O2, like so. So this is hydrogen peroxide, H2O2. So the reason it's called hydrogen peroxide is that whenever you have a single bond between 2 oxygen atoms, like this here, that's known as a peroxide bond. So this truly is just a peroxide bond with hydrogen attached to it, so hydrogen peroxide. So hopefully you can understand how you create this. All we need to add on to here is just a proton, and then these 2 electrons here can form the single bond between the hydrogen atom and the oxygen. So the positive charge of the proton would neutralize the negative charge of this oxygen. So that will create this part. And then you just need to add a hydrogen atom onto this side, so a proton and an electron, to create a single bond between this oxygen and this hydrogen. And that will create you the hydrogen peroxide. And let me just move this up again. I'm failing to keep it at the right level. Okay, so this is hydrogen peroxide here. So superoxide will spontaneously turn into hydrogen peroxide molecules. It will find hydrogen atoms and protons to react with and will become hydrogen peroxide, which is another extremely reactive molecule. So that will also react with all sorts of biomolecules and cause problems for the bacterial cell inside the phagosome. Finally, hydrogen peroxide can also be split down by another mechanism that is occurring in the membrane of the phagosome. So this doesn't occur spontaneously. It requires catalysis by another enzyme in the membrane of the phagosome. And it can be split down into hydroxide radicals, or hydroxyl radicals, I should say, rather. So you can split hydrogen peroxide into hydroxyl radicals by breaking the peroxide link, breaking this bond, sending one electron back to each oxygen, and then you'll have that unpaired electron on the oxygen, and that creates you hydroxyl radicals, which is another very reactive oxygen species. Okay, right. So here then, these are the three major reactive oxygen species that you're going to be synthesizing in the phagosome and are going to be using to poison the bacterium inside the phagosome. Superoxide, hydrogen peroxide, and hydroxyl radicals. Let me now just talk about reactive nitrogen species, RNS. Okay, so substitute the oxygen now with nitrogen, and that's what we're now discussing. Okay, so how do you create reactive nitrogen species? Well, the starting point is to synthesize nitric oxide. So on the outside of the phagosome here, you recruit an enzyme known as the induced nitric oxide synthase, the INOS enzyme, also known as NOS2. So this is the induced nitric oxide synthase. So this is an enzyme that the macrophages start producing when they have been activated by their pattern recognition receptors, and they send it to the outside of the phagosomes. And this enzyme will start producing nitric oxide. Now, nitric oxide is a free radical, however, it's a very unreactive free radical, an incredibly unreactive free radical, considering that it is a free radical. And it's not actually considered a reactive nitrogen species in itself, but it's going to react with superoxide to give us a very reactive nitrogen species. So, induced nitric oxide synthase then, it's going to start producing us nitric oxide. So let me just show you the electronic structure of nitric oxide. So nitric oxide has a double bond between the nitrogen and oxygen, like so. So if I draw a bigger diagram where I can actually draw the electrons on here. Okay, so we'll show the electrons of the oxygen in red. So of course oxygen has 6 outer shell electrons, and they're going to be like this, 4 in lone pairs and 2 in these 2 double bonds. And then nitrogen has 5 outer shell electrons, so it will have 2 in these 2 double bonds, it will have 1 lone pair here, and then it will have 1 unpaired electron. So remember, nitrogen likes to form 3 bonds overall, not 2 bonds, which is what it's formed in the case of nitric oxide. So this is a molecule of nitric oxide, and we would write this NO, and then we put a dot in front of the nitrogen to show that the free radical is on the nitrogen atom. Okay, so the induced nitric oxide synthase around the outside of the phagosome then is going to start synthesizing nitric oxide free radicals. Now this is, as I say, not considered a reactive nitrogen species, because although it is a free radical, it's actually incredibly unreactive for a free radical. However, nitric oxide can go into the phagosome, and in the phagosome it will meet a huge number of superoxide molecules, and nitric oxide can react with superoxide molecules. You see, nitric oxide has an unpaired electron, superoxide has a free electron. These can join up and form a bond. So what you can actually end up with is, here's the superoxide molecule, here's the negative charge, here's the free radical. This can join with the nitrogen of a nitric oxide which had an unpaired electron here, and then you'll have a double bond onto the oxygen there. So this is the molecule that you can overall get by joining a superoxide molecule, which is this portion, with a nitric oxide molecule, which is this portion here in orange. And this molecule is actually extremely reactive, and it's known as a peroxynitrite molecule. So this is peroxynitrite. So it's an extremely reactive molecule, and it's the classic example of a reactive nitrogen species. So this is going to react with all sorts of biomolecules of the pathogen in the phagosome, or the phagolysosome rather, and cause damage to that pathogen. Okay, right. So, to summarise then, this is the process of phagocytosis. So when the Mycobacterium tuberculosis comes into the alveoli, the alveolar macrophages and the alveolar dendritic cells are going to be engulfing the Mycobacterium tuberculosis cells into phagosomes. They're then going to be commencing the killing process. They're going to be bringing in the lysosomes and fusing the lysosomes with the phagosome, releasing lytic enzymes onto the Mycobacterium tuberculosis in the hope that that will break down the biomolecules. They're also going to be starting to poison the Mycobacterium tuberculosis by producing reactive oxygen species and reactive nitrogen species in the phagosome, and those will react with all sorts of biomolecules of the Mycobacterium tuberculosis and hopefully help to kill the actual cell. Okay, that's the process of phagocytosis. Unfortunately, in the case of Mycobacterium tuberculosis, you struggle to actually get this to work. You see, the Mycobacterium tuberculosis resists the processes inside the macrophage incredibly well. It resists the fusion of the lysosomes with the phagosome. It sets up pathways to prevent the lysosomes fusing with the phagosome. It also stops the production of the reactive oxygen species and reactive nitrogen species. So often inside a lot of the alveolar macrophages that attempt to phagocytose a Mycobacterium tuberculosis, it does not go very well at all, and instead the Mycobacterium tuberculosis survives in the phagosome, doesn't get killed, and do you know what it can even start to do? It can even start to divide inside the phagosome. So let me just go over the page, and I'll now draw you a alveolar macrophage that has become full of Mycobacterium tuberculosis cells, which have made themselves at home inside the phagosome and started dividing. So here is this phagosome, and now we've got loads of Mycobacterium tuberculosis cells inside the phagosome. So this is often what will happen. Now, some of the alveolar macrophages and some of the dendritic cells will successfully manage to break down the Mycobacterium tuberculosis that they engulf. However, most of them will fail, and you'll now actually get division of the Mycobacterium tuberculosis inside the phagosome, and this is the result, that you'll get loads of Mycobacterium tuberculosis inside that phagosome, and this can actually lead to the sentinel cell bursting open and releasing the Mycobacterium tuberculosis all over the place, and then the cycle will repeat itself. All the cells will then go off, find new phagocytes, get phagocytosed again, and you'll gradually be getting more and more Mycobacterium tuberculosis cells inside the alveoli of the lungs. So the population is not under control at all. The alveolar macrophages and the alveolar dendritic cells, they are absolutely fantastic. Okay, a lot of bacterial cells, if they were to come into the alveoli, they would be annihilated by the alveolar dendritic cells and the alveolar macrophages. Not Mycobacterium tuberculosis, I'm afraid. It goes a little bit out of control. The alveolar macrophages and dendritic cells cannot handle this. The one good thing to say is that Mycobacterium tuberculosis has an incredibly slow division time. It takes around 24 hours for a Mycobacterium tuberculosis cell to actually divide. Compare that to certain other bacterial species such as E. coli, which can divide every 20 minutes, and that does seem incredibly slow. Now that's very good for us, because that means that although the alveolar macrophages and the alveolar dendritic cells are failing to control the Mycobacterium tuberculosis population, and the population is going up, it's not going up very fast, and we have time to actually respond to this before it gets completely out of control. Okay, so what we've overall seen so far then is we breathed in this Mycobacterium tuberculosis, and the first line of defense that we attempted was to get the alveolar macrophages and the alveolar dendritic cells to clear the bacteria from the alveoli. That would have been brilliant. If they could have just cleared it away, then no problems would have been caused. However, they have failed to do that. Mycobacterium tuberculosis gets the better of those cells, and it actually, in most cases, ends up surviving inside the phagosomes and then dividing inside the phagosomes, breaking the phagocyte open and then spreading around. What's now going to happen is that the alveolar macrophages and the alveolar dendritic cells are going to start calling for help. So they're going to start releasing pro-inflammatory mediators. And we'll discuss those pro-inflammatory mediators and the inflammatory response they cause in the next video. What I just want to say before we end this video is this will all be extremely relevant to sarcoidosis. So follow the story. It is extremely relevant to sarcoidosis. One of the major theories of sarcoidosis is that it is exactly just tuberculosis. So do go along with this story. Don't get fed up of it thinking, I'm not interested in all this. I want to know about sarcoidosis, not tuberculosis. To understand sarcoidosis, the best thing to look at is tuberculosis, because the two are very similar in histological appearance. Okay, so see you in the next video.